Hello and welcome back to Microbial Concepts. So in this video we are going to discuss about isolation of acidophiles. Okay, another example of extremophiles, and you have this in your masters. Okay, masters practical. So in this video we will be discussing about acidophiles, the sampling environments, how to do sampling, the protocol, the Viva questions, etc. So do watch this video till the end. So now let's revise what do we know about acidophiles. So we know that acidophiles, they thrive in highly acidic environment. How acidic? The pH level is 3 or below. Okay, that acidic. Now they have evolved with unique mechanisms to survive and flourish in these highly acidic environments. We will discuss those mechanisms. Okay, so for now you remember that they include a variety of bacteria, archaea, and some eukaryotes. Here are a few examples you can see here. So bacteria, for example, you have Acidithiobacillus ferroxidans, lactobacillus species. In archaea we have sulfolobus, ferroplasma. In case of eukaryotes, there are few fungi. Here I have listed this one, that is Aconitum cyclophyllum. Okay, easy to remember. So at least try to remember three examples of acidophiles because you may get a question in your Viva exam. Now about adaptations. So adaptations, they have evolved, adapted to the acidic environment throughout many years, and these are the adaptations that we can see. The first one is cell membrane stability. Now, some may have thick walls or modified lipid membranes as they are less permeable to the protons, okay, H plus ions. These, they prevent the acids from entering into the cell and disrupting the internal pH balance. Now, you know, in case of acids, the H plus ions are more, right? So, the pH, if you study the pH scale, the high acidic environments, that means the more H plus ions, and in the environment if there are more H plus ions present, then the cell have to adapt to certain kind of a mechanism which will prevent the um, flow of H plus ions into the cell. Next is proton pump. Now they have highly efficient proton pump that actively expel excess protons from the cell, maintaining the neutral intracellular pH. Then the buffering system, they produce or accumulate some compounds that buffer the cytoplasm of the cell against the change in the pH, such as they have amino acids or organic molecules. Okay, they work as a buffering system. Then DNA and protein stability. Now, DNA binding proteins and other proteins, they are adapted to function optimally at low pH levels. Okay, and they have their structure intact, active. Okay, then alternative metabolic pathway. So this particular adaptation, it means that acidophiles may utilize metabolic pathways that are less sensitive to the acidic conditions, okay, such as sulfur oxidation or iron reduction. Then energy conservation. Adaptations in energy metabolism help acidophiles efficiently generate ATP under low pH conditions. Okay, so these are six adaptations. There are more, but I have listed these six as they are easy to remember and understand. Then why we study acidophiles? They can live in their acidic environment, they can flourish. Why to study them? The the acidophiles, they have ecological and industrial importance. Now, what is their ecological role? Ecological role is in nutrient cycling and environmental remediation. Now, nutrient cycling means they contribute in nutrient cycling by breaking down the organic matter and then participating in biogeochemical cycles of sulfur and iron. Environmental remediation is bioremediation. I guess you know the term, the cleanup of a given site, okay, that is highly contaminated, say with metals or any organic acids, okay. So we clean up that particular site by neutralizing the pollutant and recovering the valuable metals. Okay. Then industrial applications. First is bioleaching. So bioleaching means acidophilic bacteria, uh, for example, like Acidophiles. They are employed in bioleaching to extract metals like copper and gold from their ores, providing an environmentally friendly alternative to traditional mining methods. Okay, so if you are completely unaware of what the bioleaching means, already a video is available on my channel. You can watch that. Okay, even for bioremediation, there are two videos available on my channel in two parts. You can watch those videos as well. Then next is about biotechnology. Now, biotechnology, it simply means industrial importance of the enzymes that these acidophiles they produce or they use. We can uh, exploit them and we can use them for our purpose. Okay, industrial in various industrial processes. Now enzymes of acidophiles such as acid stable proteases, lipases, they are used in various industrial processes, in food production, pharmaceuticals, biofuel development. So we can extract those and we can use those. Now about uh, sampling environments, okay, because it is not easy to find a naturally acidic environment, okay, and get the sample. Now you need to know about what are the natural acidic environments present for the sampling purpose. Now first is acid mine drainage. uh, you can find a abandoned or active mining site if possible nearby. You know about it, the area which is exposed with sulfide minerals, okay, kind of that location. Now acid mine drainage occurs when sulfide minerals like pyrite they are exposed to air and water, resulting in sulfuric acid production and metal rich waters. Now, these environments are highly acidic, often with pH levels as low as 2 or lower, and are rich in iron and sulfur compounds. Now, sampling tip is be careful because you are dealing with acidic environments. So, collect the water sample from the drainage stream, or you can gather the sediment sample. Now, while sampling, it is important that you take your precautions. You wear hand gloves, goggles, then mask, okay, and you use sterile sampling container or sterile sampling bag. Even sampling for taking the water or scraping out the sediment, you need sterile spatula or a scooper. Then next is sulfuric hot springs. Now, hot springs are high sulfur content, which content produce sulfuric acids, creating extremely acidic or hot environments. These springs they often have visible steam and distinctive sulfur odors. Okay, so you can look for hot springs nearby and check whether it has sulfuric acid in it, whether the pH of water is acidic or not. Hmm? Collect water sample directly from the spring. Sample microbial mats or sediments around the spring at the spring edge if you see anything. Then next is peat bogs. Now, Sundarban mangrove forest in India is a best example of peat bogs. What are these? Now, peat bogs are wetland areas with accumulated organic material. Okay. Now, the accumulated organic material is peat that is acidic due to the low decomposition of plant material, and pH in these environments can be below 3 or below 4. Okay, you need to check that. Then the sampling tip can be collect water samples from bog pools, or you can gather the peat soil samples from the surface and subsurface levels layers. Then acidic lakes. Now there are naturally acidic lakes are not present, but industrially acidified lakes are present. That means Water bodies which are contaminated with the industrial effluent which has um, acidic levels of as low as 1 and 2. Okay, uh, or they can be due to volcanic activity or acid rain as well, but these are very rare and not I am not aware if they are present in India. Okay, means acidified lakes due to volcanic activity and acid rain. Okay, for that point I'm talking. The sampling tape is you can collect water samples from various depths. Okay, and it is very difficult actually when it comes about collecting the water because the pH is acidic. So I don't want you to burn your hands with acids, okay, get corrosion. So please be careful here. Then decaying organic matter. So for forest floors with abundant leaf litter, compost heaps are the locations of uh, decaying organic matter which you can use as sampling. Now here decomposing organic matter in forest compost heaps can create localized acidic environments. Okay, the production of organic acid during decomposition can range from 3 to 5. Now collect soil samples from underneath decaying leaf, gather the compost samples from different stages of the uh, heap, okay, where decomposition is going on. Okay, that can be done. Now, what are the general safety guidelines? First, I already told you wear appropriate protective gear, including gloves, goggles, okay, uh, even the mask. Be cautious around hot springs and industrial sites. Use sterile containers, tools to avoid contamination. Ensure that sampling is done properly, it is labeled properly, date is mentioned, location, and pH is also mentioned because you need to proceed accordingly in your lab. Then environmental considerations. Minimize environmental impact by taking only necessary amounts of sample. Okay, do not mess up with the site. Then next is protocol for the isolation. The isolation of acidophiles, it involves sampling from the acidic environment. So we have done that already. Now you need to use appropriate media and you need to grow the sample, okay, grow the microorganisms from the sample and then you identify it. Now samples from suitable environments. For that we need sterile sampling containers, we need pH meter or pH strip. So pH strips are convenient to carry and use, so I will recommend use pH strips. Protective gloves, goggles, sterile spatula or scoops, and cooler with ice packs for transport. If the location is far away, you need a cooler with ice pack to transport your sample to your lab. Okay. Then procedures. Identify a suitable environment. You select a site with naturally acidic conditions. You measure the pH with pH strip. Confirm that the acidity of environment is of pH 3 or below. Then you collect the sample. Okay. For water sample, you fill the sterile bottle, leaving some air space at the top. For soils or the sediment, use sterile spatula to collect and place them in sterile bag or tube. For any biological films or mats, you scrape them using a small section of and you fill them in a small sterile container. Then you label them properly. You put on the date, location, and the pH, and then you take them to your laboratory. Now about the media, different medias can be used for isolation of acidophiles. Now the material needed is for, for media preparation, distilled water, agar powder, then acidic components like sulfuric acid, hydrochloric acid to adjust the pH of your media, okay, and the pH meter to check uh, the pH, then sterile petri dish or flask and autoclave. Now, common media which are used for acidophiles are modified acidithiobacillus medium. So now this one is for acidithiobacillus peroxidans. You can see the components are given here and the pH uh, is adjusted to 2.5 with sulfuric acid. Then next one is low pH yeast extract medium. Now here you can adjust the pH with hydrochloric acid to 3. Now prepare media. Dissolve appropriate amounts of nutrients in distilled water. You carefully add sulfuric acid or hydrochloric acid to adjust the pH of to the desired levels. Okay, and then you sterilize. Okay, so before sterilization, you need to adjust the pH. Okay, now you autoclave your media at 121 degrees Celsius for 15 to 20 minutes. Then you pour plates, slants, whatever you want for your practical, and you prepare your sterile media. Then next one is sulfolobus medium. Now the purpose is it is used for isolation and growth of thermoacidophilic archaea. Now thermo means high temperature. Okay, so sulfolobus species which grows at high temperature, high acid environments. For those particular samples, those kind of samples, you can use this particular media if you are targeting for sulfolobus species. Now composition is given here. Here also you can see you adjust the pH to 3 with sulfuric acid. Now mix the components in distilled water, adjust the pH to 3, and then you sterilize by autoclaving. Then we have modified 9K medium. Now isolation, that means it is used for isolation of ion oxidizing acidophiles, often used for Acidithiobacillus peroxidans and related species. Here also composition is given. You can see. Then you adjust the pH to 2.5 with sulfuric acid. Okay, same. You dissolve ingredients, you adjust the pH with sulfuric acid, and then you autoclave. Then next is tryptone soya broth, that is TSB. This is adjusted for acidophiles. Now, general purpose is for isolation of various acidophiles. Okay, this is not particularly specified for any particular microbe. So composition is given here. You can go with this one. Okay, if you are your purpose is general purpose you have for isolation of acidophiles, you are not targeting any particular acidophile. Then same here, you dissolve the components in distilled water, adjust the pH to 3 with hydrochloric acid, and then you sterilize. Next is about cultivation. So once you have done sampling, the media is prepared, you proceed for cultivation of acidophiles. Now sterile inoculating loop pipettes, incubator which is set to desired temperatures. This is what you need. Then how to start? So you need to, for example, if you are using water, okay, as a sample, you need to do serial dilution. Okay, here I will say till 10 raised to 5, 10 raised to 6 is okay. No need for going till 10 raised to 8, 10 raised to 9. So here you can do serial dilution. You can also check directly with the sample. You can pipette out 10 microliters of the water directly to a plate. You can do spread plate and check what amount of microbes are present. As here we are just targeting to acidic microbes, so the chances are your sample will not be too much crowded, so no need of too much serial dilution. Okay, so for even for soil samples or sediments, you can proceed with serial dilution. You can pipette out last two serial dilutions, and then you can spread plate them or pour plate. If you want pour plate, you can go with that as well. So you can do streaking, pour plate, spread plate, anything that you're okay with, and then you incubate your plates. Okay, now after isolation, okay, that means streak for isolation or pour plate or spread plate. What is needed? You need to first observe the plate for isolation, mark colonies, and then you first do colony morphology. Okay, you note down all the colony morphology of the isolated microbes. Then you start with identification. Now, first thing you do is gram staining. So you need a good microscope with gram staining kit. So after doing that, you go for biochemical test. For this, you need to refer Bergey's Manual of Determinative Bacteriology. And if possible, or if you are doing research work and you have fundings for um, uh, next gen sequencing techniques, you can go for 16S rRNA gene sequencing or any other test through which you can sequence the gene of given acidophile and you can confirm its genus and species. Okay, now this is the step-by-step procedure from for identification of acidophiles, right? So I hope this is useful to you. Now, here are a few examples. I will rush through these slides because our video is getting too long. So you can pause, you can read the type, characteristics, habitat, okay, significance, okay. So I want you to know properly at least 5 examples of acidophiles, okay, because it is important for your Viva exam. So Acidithiobacillus peroxidans, Sulfolobus acidocarduris, then Ferroplasma acidermanus. Then we have Lactobacillus acidophilum. Okay. So now towards the Viva questions. So depending on the significance, depending on their environment, you may get or the sampling techniques, you may get different kinds of Viva questions. But I have here listed 6 to 8, which are very important, and I feel um, I should provide answers as well. So very first is what are acidophiles? So we have seen this in our first slide that these are the microbes that thrive in highly acidic environments with pH 3 or below. Name 2 common habitats where acidophiles can be found. So acidophiles are commonly found in acid mine drainage or sulfuric hot springs, okay, or any habitat that you have used for sampling, you can mention that. Then why it is important to maintain an acidic pH in media for the isolation. So in acidic pH in the environment, it mimics the natural environment of the acidophiles, thus promoting their growth and while inhibiting non-acidophilic organisms. Then what is the role of Acidithiobacillus peroxidans in bioleaching? Now I have discussed this one, so I am mentioning this question. So Acidithiobacillus peroxidans, it oxidizes iron and sulfur, facilitating the extraction of metal from the ores in bioleaching process. Okay. Then how does Sulfolobus acidocarduris adapt to its environment? So it is thriving in both high temperatures as well as low pH, right? It is thermoacidophile. Okay. Thus, it lives in high temperatures and low pH and it oxidizes sulfur to sulfuric acid. So this is how the Sulfolobus acidocarduris it adapts or it has adapted to the environment. Now describe one method for identifying isolated acidophiles. So traditionally, if you are doing this for practical in your college, you will go for Bergey's manual, okay, for of determinative bacteriology for identification. You doing the biochemical test. So here you do identification till genus level. And one method is 16S rRNA gene sequencing, which involves amplifying and sequencing the 16S rRNA gene to compare with the known sequence in the database. Now, why are proton pumps important for acidophiles? Now, proton pumps, they expel excess protons from the cell, okay, to outside, maintaining the neutral intracellular pH despite the acidic surroundings. Now, how do acidophilic bacteria they contribute to environmental remediation? Now they detoxify acidic or metal contaminated environment, neutralizing pollutants and recovering valuable metals. This is how they contribute to environmental remediation. So this was all about isolation of acidophiles. So I hope this video is useful to you all. Give a thumbs up. Do subscribe to my channel. Do like these videos and do share my videos with your friends. Thank you.